you. Bow your heads, please.
Thank you, God, for this day, Lord. I pray that we can look at the bread molds and all the molds, and that we can find the yeast and everything, and that we can have a great lesson. Amen.
Amen. Thank you. That we don't pass out from the smell. Okay. No, that would have been the bacteria lab, right? Okay. All right. We are learning about fungus and fungus. Fungus among us. Anyway, we learned that unlike a plant, this part on a plant would be a root, but that's actually the main body of a fungus, which is called the what? Go ahead.
Mycelium.
Mycelium. Remember, the mycelium is the part that's embedded in the food, and that is actually the main body of the fungus. Now, it has very strange thread-like filamentous cells. What are they called? Everybody. Hyphae or hyphae. Very good. And so those are called hyphae. And the mycelium is made up of what kind of hyphae? A specific kind. Rhizoid. I hear somebody back there saying it. Rhizoid hyphae, right? Okay, very good. Some of these cells, we said, were separated, and some of them were not separated. If they were separated, we called them septate hyphae, and if they were not separated, those are septate. See the separations? Otherwise, they were called non-septate hyphae. And I said that unlike any other kingdom, in fungi, we see that the cytoplasm actually flows between the different cells, which is just weird. And so they are actually have an interchanging of the cytoplasm between these cells. And then that's just showing you hyphae, the filamentous type of cells. This is the mycelium. So I know that's a mushroom, but what, as far as a fungus is concerned, what is this kind of body called on a fungus?
Fruiting body.
Fruiting body. And what is the purpose of the fruiting body?
Reproduction.
Reproduction, that's right. It's to have spores be spread so that new fungi can actually be produced. And so I'm going to go back to the first picture. It's yelling at me. Did you hear that? These are actually sporangiophores, aren't they? And let's see, these particular ones look like they don't have enclosures, so they would be candidiophores because they don't have enclosures. And you can see the little spores. They kind of look furry, don't they? Right? They kind of look furry. And so these are aerial hyphae. Remember that if they're not embedded in the food, like the mycelium would be, like the rhizoid hyphae, then it can be to absorb oxygen, or it can be for making a stolon so that it puts down a new mycelium, so it's asexual reproduction, or it can be for the purpose of producing a sporophore, which would make it either a sporangiophore, which is the spores in an enclosure, or if the spores are not in an enclosure, it's called a candidiophore. Oh, aren't you excited about that? Anyway, who knows? You might be a fungus person. I never know. You're going to be looking at bread mold under the microscope today, and I'm going to remind you of this when we do the lab, but the first time you want to look at them, you do not want to put them underwater or a slide cover, because if you just put a little piece on your slide and then look at it under the microscope and do it real carefully at the edge, it's going to look like a little forest out of Dr. Seuss's books, okay? So it'll look very, very cool. So I definitely want you to see that, and then it suggests to you to go ahead and put a drop of water on it and squish it down under a slide cover and look at it, and you're welcome to do that too, but you don't have to do that. This is what it looks like when it's squished, just so you know. And this is the sporangia, which is the spore casing. So this would be one that was in an enclosure, and this is bread mold. This shows you with the candidiophores, you know, those are with the spores being exposed. There's your stolon, which is in between, remember, and then a new fungus can grow out of that, so that's asexual reproduction. That's what we're hoping to look at today. The bread molds, the little forests under there. And remember, we talked about parasitic hyphae, which actually go into the host and take nutrients out of it, and those were called haustoria. And we'll talk about that more in a minute. We already looked last week at Chytridiomycota, which, remember, if you'll look over on page 103, excuse me, we divided kingdom fungi into the different phyla by how they do their sexual sexually reproduced spores. And so Chytridiomycota made spores in the basidia, and they're called basidiospores, and they're club-shaped cells. And so we looked at mushrooms, and I had a shelf fungus for you to look at last week, and Eric was kind enough to bring in puffballs for us last week, and I actually brought those back in case anybody didn't get a chance to look at those. I have them back there, so you can see them still this week. And then the next phylum was Zygomycota, and Zygomycota actually produces the sexual spores in the asci, which are little sac-like structures, and they're ascospores. And so you see the little sac structures here. And then, let's see, the next one is Zygomycota. Zygomycota, actually, we're going to cover today, and that would be bread molds and actually fruit molds, which I brought some pineapple mold. If you guys didn't bring any fruit mold, I do have some. And then the next listing there, I'm still on table 4.1, I'm just going over it ever so briefly, is Chytridiomycota, and these actually have flagellated cells. So what do you know about their environment if they have flagellated spores? There's water involved, because otherwise what good would it have of flagella? It wouldn't do it any good. So that's Chytridiomycota. Imperfect fungi are the ones that we really don't know how they make their sexual reproduction of spores, and they're just assuming that they do, and we're going to come back to that. And then there was Microscopic, which are slime molds, and some people put them down as in protozoa, in protista, as protozoa, because when they're feeding, they tend to look more like protozoans. But when they reproduce, they make little sporangiophores, and so sporophores, I'm sorry, and therefore we've classified them here as fungi. Okay, so and they told you it might go either way. And then we looked at the details on the reproduction of the mushroom last week. If you looked at the gills last week, you saw that in the gills there are these elongated cells full of spores, and those are the basidia, and they're full of basidiospores. And once again, we talked about that there's a good micrograph showing you the basidiospores in the basidia. And that's at a very high power. I just want you to know that. We went through that the life cycle of the mushroom, and you've got that in your book. But once again, it's the mycelia actually come together, and out of where the two mycelia come together and fuse, you have this new hyphae grow, and that's what's going to puff up into the mushroom when it comes out, and that's strictly for the purpose of us eating it and the spores being distributed. That's its purpose. And then those will produce new fungi. Once again, the puff ball, if you push on them, it puffs out. I know, pretty gross, huh? The shelf fungi can be very, very pretty as fungi go, in my opinion. All right, so that's your shelf fungi. And then we talked about in basidia, there were parasitic Chytridiomycota, which the particular ones we talked about, this was wheat rust, and it has a really intense life cycle, but within that life cycle, it requires a secondary host. Which was the secondary host? If you got rid of it, and it helped, what was the secondary host?
Barberry bush?
Right, the barberry bush. And if you could get rid of the barberry bush, then it would stop this life cycle, and so it wouldn't be able to destroy the grain crop. And then this was smut. This is what the fungus smut looks like, and that destroys many grain crops. And then we looked at Zygomycota last week, and those were your sac fungi. And so remember, the asci is the cells that are in here, and they make the ascospores, and that's what a close-up, isn't that lovely? They stained it for us. They're not that color. I just want you to know, okay? They're pretty, but not that pretty. And so those are the ascospores. The morel, which some people eat these, I don't think I ever have. You'll have to forgive me, but gourmet, I'm not this level. This is what that mushroom that smelled like death looked like to me. and yet it's in a different phylum, so I'm not sure. I would have guessed that was a morel because of this sponge-looking thing. And it said that some of these can be eaten and some of them can kill you, so you have to be careful and not eat the ones you're not supposed to, which means buy them at the store. And then we did yeast last week, and remember yeast, and we're talking bread yeast here. They bud. These are single cellular, one of the exceptions in this kingdom, and they actually grow a new one on the side, completely fully grown before it pops off. And we did see a couple budding. Remember last week I showed them to you on the microscope? And then this was, let's see, pretty sure that was wheat rust. Pretty sure. Yep. And was that ergot of rye? That's what that was ergot of rye. And the ergot of rye was in Zygomycota, and that was actually historically what destroyed Peter the Great's efforts, because he apparently got ergot of rye and his people started dying from eating it. So that can be a big problem. And then there were fungi that destroyed the American chestnut tree and the elm tree, chestnut blight, and the Dutch elm disease, and that just shows you Dutch elm disease, what it looks like, where it's got half the tree has the fungus, and so it can actually destroy these trees. And that's the American chestnut tree, and they are now gone, although they are genetically trying to re-engineer them and bring them back. It's a transgenic American chestnut tree. So that brings us to what we're doing today. Now I'm going through this quickly because I really want you guys to have as much time as you can in your lab. This is Zygomycota, and they produce zygospores. I don't know about you, but I think it's awfully nice that Chytridiomycota produces basidiospores, and Ascomycota produces ascospores, and Zygomycota produces zygospores. They made it easy for a change. Okay, I don't know about you, but I was thrilled about that part. Now, it defines for us a zygote here, and it says the result of sexual reproduction when each parent contributes half of the DNA necessary for the offspring. And my Katie Frady's easy definition for a zygote is a fertilized egg. And in general, that's what a zygote is. Once the egg and the sperm come together, you're no longer an egg, you are a fertilized egg, so now you're a zygote. But in this case, it's actually talking about when these two hyphae come together and they fuse to make something that has DNA from both of the original hyphae. That's considered a zygote. Okay, so it's just when you have DNA from two different entities come together in a new entity, and that's what a zygote is. And so in this case, the two types of molds that are in Zygomycota are bread molds and fruit molds. That's what we were told about. And the bread mold, see, bread mold and fruit mold. So did anybody else bring fruit mold? I brought a piece of pineapple. You did also. What'd you bring?
Strawberry.
Strawberry, good, because they get good molds going on. And so if you look at it under the microscope on 40 power, look like this. If you've got a big, try to get the smallest piece you can under the microscope, obviously, because otherwise it's just going to look like a black mass. But when you if you wanted to go to 100 power, go to the edge of the mass, and you'll have a better chance of seeing it if you look at it at the edge. Okay, and then you might actually see the mycelium and the stolons going from one to the other, and you'll definitely see the spores up in here. So let's see, what else? And it pictures that there for you. Oh, they have 3 different forms of reproduction. The bread mold has 3 different forms of reproduction. The asexual through the stolon, the sexual from the sporangia that releases the spores. Excuse me, that's asexual also. And then the sexual is when the 2 mycelium come together and fuse, like in figure 4.8 on page 113. When the 2 mycelium come together and fuse, the hyphae, then that produces a zygospore, and then that zygospore will grow into a whole new mycelium and a whole new bread mold. Aren't you excited? Then we have new bread mold, baby bread mold. Okay. So they can spread that way. And then once again, the fruit molds can also have those 3 forms, the 2 forms of asexual reproduction, and then the 1 form, remember the mycelium with the stolon, or just doing the spore from the little guy sticking up. Or they can actually have the sexual reproduction where the 2 come together and fuse and grow a new one from that. So that's all, let's see, that's all we were going to do on that one. So on page 114, 411, a bread mold forms a stolon for reproduction. Is it reproducing sexually or asexually? You guys tell me. Stolon is always Asexually, thank you. Stolen is always asexually, okay? 412, a fungus forms a fruiting body. Is it likely to be a bread mold? No. No, it doesn't have a fruiting body, does it? It has the sporangia, a sporophore, right? Yes, called the sporangia, but it does not have a fruiting body. Thank you. All right, then we get to Chytridiomycota, which are called chytrids. I like that a lot better. It's easier to say. And actually, chytrids are just the single-celled fungi within Chytridiomycota. And so these are pictures of chytrids. And I'm not exactly sure what they look like on a macro level, like when there's a whole bunch of them that get together and you look at it and go, look, there's fungus. I'm not exactly sure what that looks like. And I'm sorry, because all the pictures I found were microscopic pictures of them. That shows you their flagellated spores. You can see the flagella right here. And I found this one interesting because this was actually out of a biology textbook. This shows you the hyphae, okay? But then this, and it's coming out of the spore. This actually shows you the flagellated spore. So I thought that was interesting. And actually there was a cool, let's see if we can do this, is that they show this spitting out these little flagellated spores. And it's literally, you can see them spitting them out, coming through, and it's pretty neat. And that's what they're showing you for about 60 seconds or so. So anyway, then that would be in a liquid environment. And then we get to Deuteromycota. Remember, Deuteromycota are the imperfect fungi, and what's imperfect about an imperfect fungi? Yes?
We don't know their sexual form of reproduction.
Very good. So what's imperfect about them is our knowledge, right? We don't know how they sexually reproduce spores, and therefore they're put into this particular phylum, which is sometimes referred to as a holding place, because if they ever figure it out, then they'll take them out of there. But then there's also that chance that God made fungi that don't produce spores sexually. That could be too, right? We just don't know. And so that's a picture of one type of imperfect fungi. And the most famous imperfect fungi would be Penicillium. And Penicillium is the fungus that makes penicillin. And when Fleming had his petri dishes, like what we just saw, see, petri dish, and he went away for vacation, and this mold managed to get in on his petri dish and grew this blue mold. He noticed that there's the blue mold on a petri dish, so you see what it looks like. He noticed that the bacteria were dead right around the mold. And he said, wow, I wonder how that mold is killing the bacteria. So he had some other scientists help him with it. This is back in the late 1920s. He had some other scientists help him with it, and they found that these molds actually, these imperfect fungi, Penicillium, produces penicillin, and that they are antibiotics that kill bacteria. Now, they don't kill all bacteria, but they kill certain bacteria, and that's very helpful. And so on the top of page 116, it defines for us antibiotics, and it says that it's a chemical secreted by living organisms that kill or reduce the reproduction rate of other organisms. And so it specifically usually antibiotics usually are directed at bacteria. All three scientists got the Nobel Prize for their work on this. And the book goes on to tell you that if you use antibiotics too much, that you end up having immune strains produced to those antibiotics. We're going to come back to that, because that's a real oversimplification. And so when we talk about evolution, we will talk about antibiotic-resistant bacteria. And they're not produced by using antibiotics. They're naturally selected for by using antibiotics. In other words, antibiotic-resistant bacteria in a natural environment, the regular bacteria will kill them. But because you're taking antibiotics and using hand sanitizer and doing other things like that, you wipe out all their natural predators. And so now all of a sudden the antibiotic-resistant bacteria, who's normally a weakling, all of a sudden looks like a superbug, because you've wiped out all its natural predators. Does that make sense? So that's actually what's happening when you use antibiotics too much. The other problem is, does anybody else know what other concern your mom has when she puts you on antibiotics? She's worried about wiping out the flora in your gut. There are natural bacteria. I got the moms back there nodding. There are natural bacteria in your gut. You want them. They help you. They produce vitamin B and vitamin K, and they do all sorts of other good things for you. So if you're taking antibiotics, it's knocking out the bacteria in your gut too, which isn't a good thing. Although I'm not saying you shouldn't take antibiotics ever. There's times that you need them, but what your mom will usually do is either give you milk with acidophilus in it, or she'll give you probiotics after you're done with the course of antibiotics, so that you're putting back. Probiotics are just little pills with live bacteria in them that are good for you bacteria. The same as good for you yogurt has live lactobacillus in it and blah, 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 blah. Those are good for you bacteria. You want them in your gut. And so if you have to take antibiotics, you want to take those kind of things to put the good for you bacteria back in your gut. That's really important. Okay, drink kombucha, do other really offensive fermented food products. Okay, I personally live on kombucha, so you have to forgive me. Just pick a flavor you like, that's all. Okay, you haven't tasted all the flavors. You can find one. You'd be surprised. You're thinking, why would I want to? Because they really do boost your immune system. They're really good for you. Over on page 117, 413, a biology teacher once said, the only thing imperfect about imperfect fungi is our knowledge of them. What does the biology teacher mean? And you already know that's because we don't know how they sexually reproduce spores. 414, in medical journals these days, there is a lot of concern about the overuse of antibiotics. Doctors think that since antibiotics are so effective, they are prescribed far too often for patients. Why are the doctors worried about overuse of antibiotics? Because it makes strains of immune microbes shoot up. I'm just going to throw this one out to you. There are dermatologists out there that want to put high school students on long-term courses of tetracycline for their acne. I personally think they should be taken out and horsewhipped. But I just am a very opinionated person. I had one tell my oldest daughter that as I was sitting there. He said, well, she needs to be on tetracycline for the next 4 years. I looked at him and I go, are you serious? Do you actually do that to teenagers and put them on long-term antibiotics? And I'm thinking, this is why people are getting cancer and dying. This guy's crazy. So there are other ways to fight acne. There are very healthy ways to fight acne, and that is not the way, because that will just destroy your guts. And you destroy your guts and the rest of you goes, trust me. Okay, they're now starting to link gut problems with schizophrenia, Alzheimer's, all sorts of issues that we never thought of. Depression, all sorts of issues we never put together with our gut. Because basically they're now saying your gut is your second brain. Because if it's not fed here, you're not getting fed. It's not like we're osmosing the food through our skin or something, right? Seriously. So your brain's being fed right here just like everything else is. So if your gut's sick, you're going to be sick. You do not want to live on antibiotics. That is just not smart. Anyway, I'll get off my podium now. Okay, Zygomycota. That is, no, I think this was still under the imperfect fungi. We have blue cheese and camembert cheese, which I purchased for you at the grocery store. We have both of them for you to look at under the microscope. What you do is scrape off this little piece of blue stuff, and it should look something like that. Personally, I have never seen anything in this classroom looking at cheese under the microscope, but that doesn't mean we should give up. So we're going to persevere. Just make it the last thing you look at in case it's not worth looking at. How's that? Okay, but we have it. And if somebody doesn't eat one, then you can take the whole piece of cheese home, because, I can't eat cheese. I'd love to, but I can't anymore. I'm so bummed. My husband won't eat that really good, smelly cheese, How many of you like blue cheese? Like, nobody? We get to the slime molds, which that's what they look like in their feeding stage. Aren't they lovely? Oh, boy. Anyway, and they say if you want to get rid of them, all you do is dry it up, because they're like under mulch and stuff like that. If it stays wet, that's where they said you could find these. And then once again, in their reproductive stage, they look more like a fungi instead of like a protozoan. Was there anything else we needed to know about them? I don't think so. And so if we go on, and this is worth mentioning, and I don't know if I got a picture of lichen. Yeah, I did. Good. Lichen, you've seen these on trees before, probably. And this is actually very cool. It's a symbiotic relationship between a fungus and an algae. The algae gives it the green color, lives inside the fungus. The fungus provides the home, the support, and then the algae provides the food. And so together, they actually, and I think I got a picture of a microscopic. Yeah, if you actually look at it under the microscope, you can see the hyphae, so you know there's a fungal interaction going on here because you can see the hyphae, and algae don't have hyphae like that. And then the green color is actually the algae in them, which is quite cool. And what's really cool is that when they make a spore to let out, it's called a Chytridium, and it actually has a spore for both the algae and the fungi in it, which is amazing when you think about it. So obviously this relationship, this mutualistic relationship, was meant to be that way. Otherwise, how did they pull off getting their spores together in the same container, right? So it's pretty neat. And then the last one on page 120, which is also pretty neat, is the mycorrhiza. And this is called fungus root. And remember, the purpose of a root in a tree or a plant is to absorb water and minerals in the soil. Well, the hyphae, this is fungus root right here. You're seeing mycorrhiza on a piece of a root. What the hyphae does is it gives the root a whole lot more surface area because it's got all these little hairs, and all those little hairs can absorb all sorts of minerals and nutrients because there's so much surface area, because they're so small and thread-like. Does that make sense to everybody? And so what ends up happening is that this actually allows the plant to benefit by getting the mineral content that is brought in by these fungus root, and then so the root benefits, the plant benefits, and then the fungus benefit because they're actually taking their nutrition out of the root cells. So even though it's called a haustoria, the fungus root, I would say this is a mutualistic relationship. Even though they put their hyphae into the host cell and get their nutrients out, they actually benefit one another. So I just wanted to point that out, and I thought this was a really good shot. This actually shows you the fungus root, how they're in the root, and then the hyphae extends out so that you can actually see that. Pretty cool, huh? So, okay, so that's the end of that. Let's go over to get ready for the test. And while you're doing that, 4:16 says, suppose a biologist were to separate the fungus spores from the algae spores in the Chytridium. Could the fungal spores develop into free-living fungus? No, because they don't have a way of making their own food. Could the spores from the algae, the alga, develop into a living algae? Yeah, because it's got a food source that can photosynthesize. And then 4:17, there are some scientists who have studied the effects of air pollution on fungi. They conclude that air pollution destroys fungi at a much higher rate than it destroys other organisms. This same scientist says that if air pollution kills too many fungi, trees and other plants will begin to die as well. Why? It'll kill what? You can say fungus root. You don't have to say mycorrhiza. It'll kill the fungus root, right? And then the plants don't have that, and they won't have the mineral to absorb the minerals that they did. Okay, let's go over to page 123. We're going to run through this ever so quickly. Extracellular, because remember you're preparing for your test. Extracellular digestion is that they digest their food outside of the cell and then they absorb it in. The mycelium, remember, is the main body of the fungus. The hyphae is the filamentous fungal cells. Do I need to go through all these, or do you guys have pretty good definitions for these already? You're okay on these? Number 2, which of the following characteristics or structures exist for the vast majority of fungi? You guys tell me. Extracellular reproduction, either say most or few. So what do you think? Extracellular reproduction, digestion, I'm sorry, what did you get? Most, good. What about stolons? Common in the majority of the fungi. Okay, it does not list the stolons as common in the majority of the fungi. So I guess that's a few, okay? How about chitin? Most. How about caps and stalks? That's a few. How about sporangiospores?
Few.
Actually, that's few. You know what? Shame on me. Let me just... The majority of fungi have extracellular digestion, chitin, mycelia, because that's the mycelium, hyphae, because those are the filamentous cells, and rhizoid hyphae, because that is the mycelia's hyphae. Everything else there would be considered in some. Okay? So I just want you to know that. Okay, number 3. Some biologists say that a mushroom is much like an iceberg because only 10% of the iceberg is visible from the surface of the ocean. What do they mean? Good. The mycelium's embedded in the food, and that's most of the fungus, and you usually can't see that, can you? Very good. Isn't that great that she was able to do that with one word? That was just really great. That was good. Okay, let's see. Then we get to number 4. What is the difference between septate and non-septate hyphae? Remember, the septate is separated, good, and the non-septate is not. Number 5, what is the function of the following specialized hyphae? Rhizoid hyphae, what's the job there?
Well, it's the main body of the fungus.
Right, it's the mycelium, so it's for food and for support, right? Good. How about stolon? Good, asexual reproduction, that's what you were going to say, isn't it? Sporophore? It's reproduction. I think it's still asexual reproduction, but let's say reproduction. We'll be safe, okay? Here, I'll tell you what. Let's see what they say. Number 5. Yep, all it says is a sporophore releases spores for reproduction. So we'll just say spores, okay? Hostorium. Food, I like it. Parasitic food, that's right. Very good. Okay. Let's see. Then we get to number 6 of the hyphae listed above. Which ones are aerial? Good. Stolon, sporophore. Good. Number 7. What is the difference between a sporangiophore and a conidiophore?
Sporangiophore has enclosure.
Yeah.
Conidiophore has no enclosure.
Right. Makes me think they're really reaching there for that one. But anyway. Number 8. Give the main characteristic associated with each of the phyla. And remember, guys, these are all distinguished from one another by how they produce their spores sexually. So what they want there is the Chytridiomycota produce the spores, right, club fungi, they produce it in the basidia, right. The Zygomycota actually, I skipped Ascomycota, they produce it in the asci, or the little sac. The Zygomycota actually make a zygospore when the two hyphae come together. The Chytridiomycota, what kind of spore do they make? Flagellated, very good. Deuteromycota, what do we got there?
We don't have any sexual.
We don't know how they sexually produce their spores. And what about Myoxymycota? Good. They look like protozoa when they're feeding, and they look like fungi when they're not. Yeah, they're really interesting. I'd say they're confused, but I think they're just confusing to us. So, number 9, describe each of the stages associated with the life cycle of a mushroom starting. Oh, yeah. Okay, you can do that at home. Number 10, what is the main difference between the shelf fungus, puffballs, and mushrooms? What did you put down for that? Where the spores are found. In the mushroom, it's in the gills. In the shelf fungus, it's up underneath the shelf. And you guys saw that last week, I hope. And then in the furry part. And then in the puffballs, it's in the puff. It's in the little thing. Okay, don't go home and take the puff out of the bathroom and shake it out looking for spores. It won't work. Number 11, what is an alternative host? And remember, that's something that the fungi needs for a part of its life cycle to survive. And it says list types of fungi that, list a type of fungus that uses one. What'd you get? Rust. Very good. Wheat rust. Okay, remember the barberry is its alternative host. Number 12, what type of fungus is best known for fermentation? Yeast. To which phylum does it belong? Good, Ascomycota, very good. Number 13, how is budding different from asexual reproduction in bacteria?
The daughter remains attached to the parent while it's growing.
Good, good. A few of you said that. It stays attached to the parent as it grows. Very good. And then name at least 2 pathogenic fungi and the maladies. You don't have to do that here, but please make sure that you can name 2 diseases that are produced by fungi. That would be helpful. And it asks for the fungi. So you could do smut, couldn't you? And what does it do? Eats the corn. Something like that. 15, describe the 3 ways a bread mold can reproduce. And so remember, that would be the stolon, that would be the spores, or that would be the two hyphae coming together making a zygospore and then a growing one. Number 16, what, and the last one was the only one that was sexual reproduction. What puts a fungus into phylum Deuteromycota? Come on, somebody over here. How do they get in Deuteromycota?
They don't understand the asexual.
Good. When they don't understand how they sexually produce spores, they put them in Deuteromycota. Very good. Number 17, what can happen when an antibiotic is used too much? It can, go ahead. Good. It kills all the good bacteria. Very good. And it leads to strains of immune bacteria. Antibiotic resistant bacteria is what we call them. I hate to use the word immune when it comes to bacteria. 18, name the genus of fungus that produces penicillin.
Penicillium.
You want penicillium. You want to know that. You want to know that. 19, when a slime mold is a plasmodium, it resembles organisms from what kingdom? Good. Number 20, what is the easiest way to get rid of slime mold? Dry it out. Very good. And what are two major forms of mutualism in which fungi participate? Right there it's looking for fungus root and the lichen. And describe each relationship and the job. And so we said that in the lichen, the algae made the food and the fungi produced, provided the home. And in the fungus root, we talked about that the fungus actually increases the surface area and pulls in minerals that the plant benefits from. And then the plant provides the food for the fungus. So in both cases, it's mutualism, isn't it? And then what's a Chytridium?
That's like substance.
Good, and it's a spore that actually holds the fungus and the algae for the lichen, right?
Yes.
You could actually say it's a lichen spore. Yeah, you could say that, couldn't you? Okay, flip back to your labs.